Professor Deborah Mayer, Executive Dean, Faculty of Science, Professor Peter Teske, inductee, Professor Bettine Janssen van Vuren, respondent, Senior Director, Strategic Initiatives and Administration, senior leaders of the university, members of Senate and other academics, to those of you who have joined us through Facebook and YouTube, distinguished guests, ladies and gentlemen, Sanibonani, Khoyanand, good evening, Tobena. It is indeed a great honor and special privilege for me to welcome you to the Professorial Inaugural Lecture of Peter Teske. As I do so, I wish to express a warm welcome to his loved ones, special guests, and his colleagues. This is indeed a proud and joyful, yet a landmark moment for all of you, for Peter, and of course for us here at UJ Higher Education in South Africa and beyond. The inauguration of professors is a public ceremony in which newly appointed professors are inducted into office by the Vice-Chancellor functionary and deliver their inaugural address. The ceremony has its roots in the medieval university and serves multiple purposes. Firstly, it is an expression of welcome and an entry for new professors joining the circle of colleagues who are already professors. Secondly, it provides a welcome for the professors to display their expertise in the discipline and showcase their research. Traditionally, in isiXhosa, we say, "Ukutweswa isitanga kwisigaba sobunjingwa lwazi." This, loosely translated, refers to assuming the role of the professor. Of course, in colonial traditions subscribed to by universities, this refers to the gown and the cap. The wise one would accept a blanket in Gubo. Once we have listened to the inaugural address, the gown, or in Gubo, denoting the professorship, will be formally assumed. We gather to bear witness to the entry of Professor Teske to the illustrious community of scholars at the university. It is a celebration of the contributions of scholars at the university. It is a celebration of the contributions to the discipline and ultimately the impact to society. Professors provide a university with its identity, character, academic legitimacy, and integrity. The inaugural lecture is a rite of passage following the confirmation of the appointment of the person as a professor. This evening, we will listen to Professor Teske as the gown goes to town. By this, I mean the power of the inaugural address is when the expertise is showcased beyond the corridors of the university and reverberates in society. It stands out as a moment of pride for the incumbent, the family, fellow scholars, the university, and ultimately society. For the German philosopher and diplomat Humboldt referred to the university as a whole community of scholars and students engaged in a common search for truth. Newman talked about teaching universal knowledge. Recently, universities have been viewed as instrumentalist, serving the purpose of the economy or utilitarian in purpose. I would hope that we can break out of these narrow conceptualizations and reflect on the university as contributing to public good. Edward Said, in an article on defiance and taking positions, offers the formulation of the ideal role of the true intellectual. Open quote, "One who commands a vast knowledge of the person's discipline, who is rigorous in the analysis of literature, who views being an intellectual as a vocation, intellectual who considers it necessary to step into the public sphere and to speak truth to power, namely to question, interpret, and understand authority rather than consolidating it. To step out of the boundaries of the academy, to connect oneself, to affiliate oneself, to align oneself with an ongoing process of contest of some sort, perhaps with the aim of improving the lot of the oppressed. The intellectual who functions as a kind of public memory to recall what is forgotten or ignored, to make the connections that are otherwise hidden, and to provide alternatives for mistaken policies. Close quote. It remains for us as a university with a Pan-African vision to derive our mandate as intellectuals and as professors. How do we embrace the role of a professor as a disruptor while continuing to be flagship carriers of our disciplines? This evening, we will listen to Professor Teske as one further step in the journey of being a professor. This is a journey which does not culminate once this lecture has been given. It is a self-reflective pause in the journey of the professor with the promise of more to enrich our minds and simultaneously contributing to the rich intellectual body of work in the discipline. Let me now invite the Executive Dean, Professor Deborah Mayer, to introduce Professor Teske. I thank you. Ria Leboga, Giabonga, Bayadanki.
Thank you, Professor Sina. Good evening, ladies and gentlemen. It is my absolute pleasure to introduce Professor Peter Teske, who will be our speaker this evening. Peter Teske was born in Hamburg, Germany in 1971 and spent his early childhood in Empangeni in the northern KwaZulu-Natal. After completing his schooling in Germany, because of the political situation in South Africa, he permanently returned in 1994, the year Nelson Mandela became president. He completed his BSc Honours in Zoology and MSc in Estuarine Ecology at the University of Port Elizabeth, now known as Nelson Mandela University. This was followed by a PhD on the population genetics and phylogenetic placement of the endangered Knysna seahorses, and this was done at Stellenbosch University. To acquire new skills in a rapidly developing research field, Professor Teske then spent several years in Australia, first in Sydney and then in Adelaide. He joined UJ as a senior lecturer in 2013 and was promoted to associate professor a year later and to full professor in 2020. Prof. Teske has been instrumental in introducing 4IR technologies to the biological sciences at UJ, including genomics, transcriptomics, and meta-barcoding. All these methods rely on the generation of large amounts of DNA data using massively parallel sequencing coupled with cutting-edge cluster computing. Professor Teske's research group uses these technologies to advance marine biological research in Southern Africa with the aim of improving the management of exploited or threatened species. By doing so, he often challenges long-established paradigms. Current research projects include genomic and transcriptomic research to understand the KwaZulu-Natal sardine run, population genomic research aimed at improving the management of the critically endangered estuarine pipefish, which is one of Africa's rarest fish, and meta-barcoding projects aimed at documenting the biodiversity of estuaries from northern Mozambique to the Eastern Cape. It is now my pleasure to introduce to you Professor Peter Teske.
Right, thanks to my colleagues for the kind introduction. The inaugural lecture is an opportunity to showcase my research. But before I do that, I will take a lengthy detour into something that I know virtually nothing about, psychology. Or you may also call it pseudo-psychology, as I'll explain a bit later. Like many of us, I spent way too many hours reading things on the internet during lockdown that are not related to my work. And while I was busy trying to find out if I might be slightly autistic perhaps, I came across this construct here, the Myers-Briggs personality types, which have been ridiculed as Chinese fortune cookie psychology. They are based on ideas by the Swiss psychiatrist Carl Jung, and they essentially assign every person to one of 16 personality types. half of them that start with an I are introverted personality types, and the other half, starting with an E, are extroverted personality types. This may be pseudoscience, but there's a large internet community that has dedicated its life to this, and I know that some universities in South Africa are in fact using this to place academic staff members. And in my case, I found some amazing matches that I'd like to share with you before I start with a more general discussion of how all of this is actually relevant to my research. So I did this online test, and I was identified as a INTJ personality type. And amazingly, this test knows a lot of things about me that I thought were unique to me, but apparently there's others like me out there. So I'm a sort of a lone wolf personality. I believe that if you want it done properly, you do it yourself. This is obviously not ideal to do collaborative science. I'm socially awkward, that's also correct, and romantically clueless. I have yet to go on a date, it's on my bucket list, but first I need to learn how to speak Portuguese fluently. I do something called the death stare. When I try to concentrate, I apparently convey the impression of malevolent pity. It's essentially, imagine it as, I should probably feel sorry for you, but I just hate you too much. That is my normal facial expression, essentially. So that causes misunderstandings. I have something, believe it or not, that is called telephonophobia. That is quite common among introverts. By the way, we hate Zoom meetings as well. And more importantly, in the context of my speech here, is that the INTJ types, even though they're extremely rare in the general population, males I think make up only 2%, females are even rarer than that, they tend to accumulate in certain places. And one of these places is science departments at universities. In other words, if you are in a faculty of science, you are quite possibly surrounded by INTJs without realizing it, or you might be one yourself without realizing it. More generally, and more relevant to what I will be talking about here, are three traits. The first one is, there is no respect for hierarchies. Social hierarchies are irrelevant. So if you call me Mr. Teske after this meeting, I will not mind at all. The professor is just a title. Then they tend to be strategists and go-getters. It sounds a bit like a contradiction. Introverts usually think a lot and then struggle to implement it in the real world. The INTJs think a lot and then actually manage to implement it. And they're considered visionaries or rebels, depending on how you want to see it. They are not afraid to step on toes. They believe that the truth is more important than being popular. And I'll show you a lot of examples of how I have stepped on other people's toes, and why that is not actually a bad thing if you want to advance science. So, if you are a little bit like this, an INTJ, it is beneficial if you come out of your comfort zone. Stop being a lone wolf entirely and acquire some more extroverted skills that will help you collaborate with people. If you are not an INTJ, some of the personality traits that INTJs have might be quite useful if you want to make a name for yourself in science. So my presentation is primarily aimed at emerging scientists. So I'm sharing some old man advice here with you. First things first, these slides are not trying very hard to be dignified. If you are an INTJ, you need to keep the extroverted people happy. One way of doing that is to do small talk. They like to do that, silly things like, hi, how are you? Fine, how are you? Fine. It's completely irrelevant, it's pointless. It's just a way to avoid awkward silence, or as we would call it, silence. And you have to smile. That is something that doesn't come easy to INTJs. You hardly ever smile, or at least not in this way, by showing your teeth, which we consider to be a sign of aggression, really. Unless you're in America, if somebody tells you to smile, then it's workplace harassment, and you can get, the person can get fired. That might eventually arrive here, but for now, try to smile occasionally. All right, now some specifics. No respect for hierarchies has certain advantages and disadvantages. So In this case, I have a typical academic hierarchy with the dean on top and the postgrads at the bottom, or of course the undergrads should be below that. This is pretty much meaningless to the INTJ. They have their own system. It's a binary system, and it divides everyone below the level of dean into either competent or incompetent. No one's safe. And the positive thing about this is if you have only a binary system, there is no room for sexism, racism, homophobia, anything else. You're either competent or not. And that way there's a lot of communication up and down this pyramid. The disadvantages is that if an INTJ classifies someone as incompetent, they might not want to deal with them, even though it might actually be quite useful for their research. For example, if an extroverted person talks too much, the introverts will try to avoid them, but having people like that on a team is actually extremely useful, as I've learned. So, how do the INTJs perform in academia? As a student, they tend to be irritating. These are the ones who will challenge the lecture. They have no respect for even a professor if they think the professor is wrong. And they also are often confused by, what the other students are fascinated in doesn't fascinate them very much. Of course, these traits are a bit annoying at that level, and that's primarily because they are low in the hierarchy. As soon as the INTJs move up in the hierarchy, the situation changes, and suddenly what was annoying in the past is becoming very desirable. So as a teacher and mentor, this is a surprising thing. They are actually quite good at that, even though they're introverted and they don't like to hear themselves talk. I've avoided teaching for a long time. I did one postdoc after the other. And then I started teaching at UJ because it is something I have to do, so that I can do research as well. I realized that the students actually like my lectures because I don't overwhelm them. I focus on a few concepts and they're discussed in detail. So the students learn something difficult, but they are given the time to do it. As a supervisor, I'm actually also pretty good, I'd say. There's no detached supervision. This is not some arrogant supervisor. If the supervisor identifies a student as competent, then that student is an equal. It's simply an equal who has less experience, and all I do is put that student on track and give advice. As a collaborator, this INTJ type is also quite good. As a team leader, they have a vision, they have worked it out in their brain. Now they go around and identify people who can help them realize that vision. And they, in that way, assemble highly diverse teams from many different scientific disciplines. As a team member, they will also be very strongly involved. There is a risk that they will take charge if the actual team leader is suddenly considered not good enough. The INTJ might take over. So be aware of that, and be aware that there will be a hell of a lot of email correspondence because Zoom and telephones are avoided. Right, now how does all this feed into advancing science? The most important point here is the idea that the INTJ does not accept the status quo. And I found that a lot of time in myself. I've seen it in many others who advance to become good scientists. So here's a quote from Bertrand Russell. Conventional people, by that he probably means non-INTJs, are roused to fury by departure from convention, largely because they regard such departure as a criticism of themselves. In science, it is necessary to depart from convention. That is what advances it. A little bit of background here on the philosophy of science. Many of our students, staff members, have done at least one lecture on the philosophy of science. Most of them considered it a total waste of time. I personally considered it very useful, especially the ideas of Karl Popper. So you may be aware of falsificationism. A hypothesis is scientific if and only if it has the potential to be refuted by some possible observation. It's very different from the social sciences where your truth and my truth is equally good, and there's no such thing as the truth. We have nature to tell us how bad or how good our hypothesis is. More important in this context of my presentation is the idea of a bold hypothesis. A hypothesis that is bold to the extent that it risks falsification. If your hypothesis cannot be rejected, then it's not a good one. And I will give you a couple of examples of how I have expressed bold hypotheses throughout my career. So I started being a person who questions science and tries to advance it in my MSc years. I learned in marine ecology at UPE, now Nelson Mandela University, that the community structure of estuarine microbenthos in South Africa is linked to salinity. An estuary is essentially a river mouth that is influenced both by the ocean and by a river. And as you can see in this figure here, these are essentially the sites in the estuary grouped according to faunal similarity. And if you superimpose salinity, you don't find much of a pattern. There's one group of oligohaline species, but otherwise, what is called the Riemann model, which works well in Europe, doesn't seem to work terribly well in South Africa. Instead, if you superimpose sediment size, you find an interesting pattern that some of these sites are in a sand zone and the other sites are in a mud zone. And this makes a lot more sense than salinity. Because you have to imagine there's a crab sitting at the bottom of the estuary. In the morning the tide pushes in, the water becomes more saline. In the afternoon it rains, the river pushes in and the salinity drops. The crab sits in the same place all the time. It does not care what the salinity is because it is a great osmoregulator or osmoconformer. And that is essentially the reason why the salinity scheme doesn't really seem to work in South Africa. Another thing that is unusual about this plot here is that everyone tended to do this for one estuary at a time. I decided to throw them all together. So this is actually a composite of 13 different estuaries, and the pattern was present in all of them. After completing my MSc at UPE, I went to Stellenbosch University, and after working on Neisner seahorses, which I will not discuss today unfortunately, I started several postdocs with the aim of connecting with my past as an estuarine macrobenthos researcher, using genetics in this case. And in those days we sequenced a lot of mitochondrial DNA. Today this would be known as DNA barcoding, which is a great way to determine species status genetically. So this example here is not an estuarine, these are not estuarine species, it's actually a rocky shore snail, a little grey one. It was known before we started this that these were two distinct species, but the genetics definitely confirmed it. So I have been accused of being someone who generally bashes DNA barcoding. That is not true. I'm actually involved in a lot of projects right now that use mitochondrial DNA. I just caution against inappropriate use of the method. So barcoding is definitely very useful. And I have an example here. This is a very simple one. This is not for IR at all. The student here, Chifiwa Matumba, essentially wasted a lot of money on sequencing a genetic marker that should not show any variation. And what you're seeing here is that even though the two snails here are not strongly differentiated based on this marker, within, at the species level, the genetic diversity is actually quite similar to that of CO1. So my main criticism coming out of this study is that molecular dating using mitochondrial DNA at the species level is a waste of time. Because the top marker evolves at least 10 times as fast as the bottom marker, and yet they have pretty much the same diversity. What we're seeing here is most likely selection effects on the top marker, the mitochondrial one. So essentially, what we're seeing is a reduction in genetic diversity as a result of stabilizing selection at the species level and increased divergence rates between the species. So in other words, this is not useful for molecular dating because this marker does not evolve in a molecular clock-like fashion at all. And with a simple study like this, we can essentially reject thousands of studies that have used this approach for molecular dating. Another thing we started to find is that species that are widely distributed along the coastline can be divided into regional clusters. Initially, at this point, I started working with Sophie von der Heiden occasionally, and we have deepened our collaboration quite a bit. And we were very interested in what is causing this. Initially we thought it must be some isolation effect. They cannot get to each other, so over time they will diverge and become new species. But I think that is pretty much refuted. Because, for example, during El Nino events, a lot of tropical, colorful fish species or crabs can establish themselves far outside the normal range. They will just not stay there for very long. Eventually, especially when upwelling starts, they will disappear again. So it seems to be the environment that is causing the divergence here, especially water temperature. This is an image that I created for a presentation in Istanbul. And I have essentially given them examples of how the bioregions in South Africa, how similar they are to regions in Europe, the Middle East, and North Africa. Our west coast is essentially similar to Norway. The southwest coast is a bit like the Irish Sea. The south coast similar to Portugal. The east coast like the eastern Mediterranean. And the northeast coast tropical like the Red Sea. If you take a fish from the Red Sea and drop it in Norway, that fish will not live for very long. And it is essentially a similar situation here. And yet you have some species that are distributed along this entire coast. Clearly, there cannot be one and the same species. There must be multiples. This genetic division essentially mirrors South African biogeography, which is based on the presence and absence of species, and which is also based on water temperature, essentially. So to show you this picture of the sand prawn again, we have clear divisions into cool temperate, warm temperate, subtropical and tropical regions here, matching South Africa's marine biogeographic provinces. Essentially, you have new species evolving here. Why exactly they're evolving in these regions, not sure. They seem to be adapting to a certain water temperature, and if they move to another region where the water temperature isn't right, they will not do very well. That was our original hypothesis. And we essentially started exploring this further. Here's another prawn. And this was one of my first major collaborations outside my immediate field. I found genetic structure in this prawn, and I approached physiologists who were doing pretty much aquaculture research. They were rearing the larvae of this prawn, and they found that the subtropical larvae cannot complete development at the lowest temperature. And since I'm an INTJ and do not really respect hierarchies, I approached the Taxonomic World Authority in this particular group and asked him to find some morphological differences, and he actually found one. There's a spike that the subtropical one has that the warm temperate one does not have. It's probably not enough to call them different species, but they are definitely on their way to become new species. This is also not 4IR. But what I'm doing next is, we have now explored this in more detail, because the temperate prawn is also further subdivided into a warm temperate and a cool temperate west coast form. Morphologically completely indistinguishable, but they seem to be on their way to become distinct species as well. So what we've essentially done is we've kept these prawns at 20 degrees. This is a temperature that both of these lineages experience all the time. And there were quite a few genes that were differentially expressed, up or down regulated. But when we put them at 10 degrees, which is a temperature typical of cold upwelling that only the west coast prawn experiences, the number of differentially expressed genes doubled. So these prawns were really struggling. And essentially this means they can establish themselves in the habitat of the other, but only briefly. Once the upwelling starts, they will disappear. And that is essentially a driver of genetic divergence. If you ask yourself, how is this baseline research useful to society, to food security, to conservation? I have a couple of examples here. This is very important for stock management, including fish or the alga that I'm showing here. This is an ongoing study. Essentially, if you over-harvest the populations shown in red here, because you think these populations will get replenished from the green area, you might be wrong. Maybe they're cryptic species. Maybe they have nothing to do with each other. And if you get rid of the one, it will not get replenished. Your stock will collapse. This is equally applicable to South Africa's fish stocks. Another application, this one is entirely hypothetical. We've only recently started large-scale DNA barcoding on South African sponges. The long-term aim is to find bioactive compounds with medical applications. Imagine the green one here has a compound that can be used to cure cancer. But you want to mass produce them on the west coast because there's more plankton in the water to filter. It will go faster. It's entirely possible that the bioactive compounds are different in that blue population. So before anything like this is done, one first needs to sort out, are we dealing with different cryptic species or different populations here that might be differentially adapted. Another example, conservation. This is an extremely rare limpet here, Siphonaria compressor. There's only two populations. The species is reasonably common in Langebaan Lagoon, but extremely rare in Knysna. This population lives in an area that is probably smaller than this room here. Now you might decide, okay, this small population is probably inbred. It needs, we need to improve its genetic health. We need to strengthen it by translocating individuals from the Langebaan population. If they are differentially adapted, what you will essentially do is introduce a competitor, or you might have a situation where hybrids will form that are not well adapted to any conditions, and you will actually accelerate the extinction of a Knysna population, which might be the distinct species. All right, back to some mitochondrial DNA bashing. The concept of isolation by distance is also something that has been used in thousands of studies using mitochondrial DNA and other markers. This is a very simple idea. The greater the geographic distance between two populations, the more different they are genetically. What a lot of people get wrong is that they're actually throwing together genetically distinct lineages that might be different species. And of course, if they are adjacent to each other, you will find a pattern of isolation by distance. But if you focus only on a single lineage at a time, essentially the pattern will be lost. This is one of our highest impact papers so far. This is the first genomic paper we have produced. On the left you see the mitochondrial DNA showing a flat line essentially, slightly negative even. So the mitochondrial DNA at the lineage level is not finding isolation by distance. On the right you see SNPs, single nucleotide polymorphisms, thousands of them. And in this case you find a completely different result, a highly significant correlation between geographic distance and genetic distance. Again, this challenges the findings of thousands of previous studies. And I'm really stepping on toes here, but I'm also stepping on my own toes, because I have also used this in the past. Alright, so here's a bold hypothesis for you. Species that are present in multiple southern African marine bioregions are in fact multiple cryptic species. This hypothesis seems to be dead in the water right from the start, because we know already there are exceptions. Many species do not show this. The question is, why? Is it possible that DNA barcoding is simply not suitable for this? And we explore this using genomic methods. So essentially, another bold hypothesis, lack of genetic structure across marine regions is an artifact of low genetic marker resolution. The mitochondrial DNA fails to find it even though it is present. Maybe because these young species have evolved very recently. So some species are quite old, you can tell them apart genetically, morphologically. Some are younger, you can tell them apart genetically. You can see the phylogeographic breaks in black here. They are quite uneven. And then there are some examples of animals that show no genetic structure along the entire coastline. And we decided to test our hypothesis by focusing on this goby here. And using thousands and thousands of variable genomic sites, we didn't find anything. But if you take into consideration only the loci that are correlated with sea surface temperature, we essentially found a pattern like this. On the left-hand side, CO1, genetic homogeneity, and on the right-hand side, the expected pattern. Just much younger than in the other species that we've studied so far. These are in the early stages of speciation. Another challenge, could it be that if you don't find genetic structure, it might be related to dispersal ability? The goby just sits on the bottom and doesn't do very much. What if you study an actively dispersing fish? Will we find genetic homogeneity, or does the pattern still hold? So here's another bold hypothesis. Actively dispersing species can move to preferred habitats, but these movements will still be affected by biogeography. In other words, we still expect a temperature effect, even though there's a lot more mixing going on. And in this case, we got really brave, and we studied a species that we know can disperse easily all over the place, and that is the South African sardine. You may know this from the famous KwaZulu-Natal sardine run, the greatest shoal on earth. In winter, essentially, tens of millions of sardines move up the east coast, and it's not entirely sure why they did that. It has been hypothesized that there are actually 3 sardine stocks in South Africa, which could explain why the west coast stock has collapsed. It has been overfished and wasn't replenished from the south coast stock. And there might be an east coast stock that mixes with the south coast stock in summer, and then moves up for spawning up the east coast in winter. Before I move on, let me explain to you how a structure barcode works. I will show that from then on. Vertical lines indicate single individuals. The first one here is part of stock 1, the second one part of stock 2, and the third one might be the offspring of the other 2. And we found this pattern. This is a bit difficult to understand. It's a lot of fish here, and superimposed is maximum and minimum water temperature at the site. So I'll walk you through it very slowly. As you can see, there are only 2 stocks. And on the south and southeast coast, all of these have almost completely black ancestry. So they're part of stock 2, there's 1 or 2 exceptions. On the southwest coast, you also find some that have that, but you also find some that have more grey stock 1 ancestry. And on the west coast, most of the individuals are mostly of grey ancestry. There's 1 exception shown with the arrow here. And these are most probably migrants from the south coast, and we found these at the warmest site. They are in the wrong place. They don't seem to like that very much. They are essentially moving to areas that are a bit warmer. And then we have the sardine run here. This is pretty much a mixed bag. But what is interesting about it is the proportion of stock 2 ancestry here is lower than on the south and southeast coast. So these are actually, these originate from the west coast. Some of these are almost completely stock 1, others are mixtures. We don't find that on the south coast, with a few exceptions. So what's essentially happening is, these are west coast sardines that are in the wrong place. They get attracted by the upwelling that starts a sardine run. They move up into what they think is good habitat. Then the upwelling stops and they find themselves in subtropical waters, in the worst possible place. At the same time, they get chased by all sorts of predators, fish, seabirds, mammals. So lastly, genetic homogeneity is rejected even for this high dispersal fish, but we have a bit of an unusual pattern here. We have only 2 stocks. In conclusion, the INTJ tendency to thoroughly explore data can help to detect meaningful structure where there seems to be homogeneity at first sight. In other words, this is sort of the advice for our students. Don't do a paint by numbers approach. Don't repeat what everyone has done before you, but try to explore the data in much more detail. Because discovering hidden patterns that are not immediately obvious takes time, creativity, and I've put this in here, discussions. This is something the INTJ doesn't do automatically, but it is definitely extremely useful. And lastly, formulate a bold hypothesis, and then before anyone can shoot it down yourself. Or try to improve it, try to refine it. And with that, I'd like to thank you very much for your attention. I would normally say I'm open to I'd be happy to answer some questions. That's clearly not possible yet. If you have any questions, you can email me. Do not try to phone me, I will not answer them. Thank you.
Thank you very much, Peter. That was a typical Peter talk. Let me start by saying that I've known Peter now for probably 15 odd years from Stellenbosch days. And in keeping in line with the presentation, let me start with an anecdote. So we had a fridge, one of those sliding door fridges in the laboratory at Stellenbosch University. And Peter would work during night times. He would never be there when everybody else was there. He was there over a weekend. You would know that Peter was there because he had a big wolf outside the lab, Cosmo. And as usual, Peter worked over the weekend. Nobody else was there. And on Monday morning, I walked in. At that point, I was managing the lab. Walked into the lab, and here's a nice big note on the whiteboard saying, "The fridge is now an oven." And Peter's reagents have been removed from the sliding fridge, and everybody else's stuff is there, been sitting there for the whole weekend. Boy, that was the day that I thought the lab was going to kill Peter Teske. But thankfully, he only arrived at night, so he was safe. And I'm hugely honoured and privileged to say that we've come a full circle, and 15 years, 20 years later, we share the lab at the University of Johannesburg here. Okay, on to serious things, because this is a serious event. The Earth we are undoubtedly experiencing a biodiversity crisis, and I think a lot has been written about this biodiversity crisis, and what is really needed is innovative and proactive responses from researchers like Peter Teske to governments, and we're really thankful to say that South Africa here is leading and is a front row player in it. And when we keep with the marine focus in terms of marine protected areas, South Africa has close to 50 marine protected areas, which covers approximately 5% of our territorial waters. This really is massive and compares incredibly well with other countries. These marine protected areas are near shore and in open waters. And in the face of these protected areas, so where habitat and species are actually protected, it's very important to understand the connectivity or the movement of marine species between these areas. This, of course, allows for optimal management and is absolutely critical to protect our oceans, both in terms of the biotic and the abiotic factors that take place there. If for nothing else, then the oceans are actually huge carbon sinks. So in this space, Peter Teske really is doing world-class research, and as he said himself, he challenges bold hypotheses continuously, essentially. Peter Teske has been in South Africa and globally a leading player in this field, and he leads by example and he leads from the front. One example of this is in 2011. He was the first author on a paper and essentially drove this paper, which was a review on the spatial patterns of marine species across the South African coast. The author line really reads like a who's who in that specific sphere of marine biology, and today that paper has more than 200 citations. Peter has an H factor of 28. I checked just before I walked over here, with close to 2,600 citations. And that really is extraordinary for someone of his age. You might say old man wisdom, but he's really not that old. He really is doing exceptionally well. And then in line with the University of Johannesburg mission to advance the fourth industrial revolution, and there again to lead from the front by advancing not only South Africa but Africa into the fourth industrial revolution, Peter Teske really is dragging a lot of people kicking and screaming with him into this 4IR sphere. He really is leading from the front in this area as well in South Africa, as he's showing you some of the examples here. He drives the omics research in South Africa, essentially in this field, and I really am privileged to share the laboratory with someone of the stature of Peter Teske. Thank you very
I now call upon the Dean, Professor Deborah Mayer, and Professor Peter Teske to receive the gown, to have the gown process completed. Colleagues, I'm sure you would let us give a round of applause wherever you are to Professor Peter Teske for a fascinating presentation and lecture this evening. I thought it was an excellent lecture. I think we learned quite a bit about the special nature of marine biology that we have in South Africa, the different geographies that you brought together. But most interesting was also the aspect of the INTJ and how this personality has in fact not only benefited you, but could benefit others. And the aspect of being able to question things, being able to reach out to individuals, breaking boundaries, both in connectivity, but also to be able to harness the power of the Fourth Industrial Revolution as it connects to the discipline of marine biology. Let us give once more, Peter, congratulations, and let's give Peter a round of applause. With this, we now come to the end of this evening's ceremony. Colleagues, friends, ladies and gentlemen, from wherever you have joined, I hope you've enjoyed this session, and thank you.
The University of Johannesburg, the future reimagined.